Before we dive into our chosen research article, let's consider the video that you just watched. It depicted a man experiencing PTSD flashbacks, a vivid example of how fearful memories can persist and disrupt daily life. PTSD highlights the lasting impact of fear conditioning. In psychology, we've learned that fear conditioning is a process where a person or animal develops a fearful response to something neutral like a sound after it's been paired with something unpleasant known as an unconditioned stimulus like a shock. For instance, imagine hearing a beep every time you touch a hot stove. You might start to feel anxious just from hearing the beep, even without the burn. But here's the good news. Fear is not set in stone. In fact, it can be reduced through a process called extinction. Think of extinction as a way to retrain the brain. By repeatedly experiencing the fear cue, like the beep, without any negative consequence, the brain learns that the cue is safe and the fear response gradually fades. Previous research has shown that extinction relies on specific brain structures. When it comes to the brain's role in fear and extinction, two key players are the amygdala and the ventral medial prefrontal cortex, also known as the VMPFC. The amygdala acts like a fear alarm, quickly signaling danger and storing fear memories. But the VMPFC works like a fear manager, gradually learning to turn down the alarm by helping control the fear response. Over time, as the VMPFC and amygdala connect more, the fear becomes less intense and easier to manage. Most of our understanding of fear conditioning and extinction comes from rodent studies. In these studies, fear conditioning strengthens neural pathways in the amygdala where fear memories are stored. For example, mice conditioned to fear sound paired with a mild shock showed increased receptor activity in the amygdala, reinforcing the memory. This heightened activity makes neurons more likely to fire in response to the fear cue, keeping the memory vivid even after the conditioning ends. Extinction is effective at reducing fear, but here's an important catch. It doesn't actually change the original fear memory. Instead, it mainly helps manage the fear response. Recently, though, researchers have been exploring a fascinating possibility. What if we could alter the memory itself? This is where reconsolidation comes in. When a fear memory is reactivated, it briefly enters a reconsolidation period where it's temporarily open to change before it stabilizes again. In 2013, Schiller and colleagues explored this, testing whether extinction during reconsolidation might offer a unique opportunity to alter fear memories directly.
So what was the specific research question and why was it important or timely? The study questioned if we can reduce the brain's reliance on fear memories by using a process called reconsolidation. Reconsolidation is when a recalled memory becomes soft for a brief time, which means it's open to change. The researchers exposed people to a reminder of a fearful memory, but without any negative outcome afterward. By doing this, they hoped to see if the brain would adjust its response to this fear memory, specifically reducing the prefrontal cortex involvement, which is heavily involved in managing fear. This study is important as it can transfer mental health treatment by directly altering fear memories rather than just managing them, offering more lasting solutions for conditions like PTSD and anxiety. It also enhances our understanding of the brain, paving the way for innovative and effective therapies in the future. Moving to the next question, what specific tasks and methods did the researcher use? The researchers used a three-step approach to create and then weaken a fear memory in a controlled way. On day 1, the learning fear stage began, during which participants were trained to associate a neutral image, a colored square, with a mild shock. This created a learned fear response to the square known as conditioning. Next, on day 2, the memory recall and extinguishing fear stage began, where participants were briefly shown the square again, but this time without the shock. This reactivated the memory of fear, opening what is called the reconsolidation window, a brief time where the memory could be changed. Lastly, on day 3, the testing fear recovery and further extinguishing stage began. During this reconsolidation window, the researchers showed the participants the square repeatedly, without the shock, aiming to reduce the fear response. This part is similar to traditional exposure therapy, but done in a way that might weaken the original fear memory. To measure whether this approach worked, several methods were used. A skin conductance response, SCR, was used to measure sweat response, which tells the researchers about a person's level of fear. By checking SCR before and after the extinction phase, they could see if the fear levels dropped. An fMRI was also used to focus on activity in the prefrontal cortex and amygdala, two brain areas heavily involved in the fear processing. They compared brain activity for participants who experienced extinction during the reconsolidation window and to those who did not. Lastly, a statistical analysis was used. They combined data from SCR and fMRI to check for significant changes. If fear response and PFC activity dropped notably during extinction in the reconsolidation window, it would suggest that this approach effectively weakens fear memory.
Okay, so we'll now discuss some of the key findings. The first key finding was a reduction in prefrontal cortex involvement. During extinction in the reconsolidation phase, activity in the ventromedial prefrontal cortex decreased. Normally, the VMPFC helps regulate the amygdala's fear response, and in standard extinction, its activity increases over time. However, this increase wasn't seen with the reconsolidation-based extinction, suggesting this method might directly alter the original threat memory instead of relying on the VMPFC to suppress fear. Next is the amygdala involvement. Both the reminded and the non-reminded condition showed increased amygdala activation early in extinction, reflecting a fear response. However, only the non-reminded condition maintained the typical amygdala to VMPFC connectivity seen in standard extinction. In the reminded condition, amygdala activity decreased over time, suggesting a deeper, more lasting change to memory rather than temporary suppression. Another key finding involves the return of defensive reactions, which is measured through the skin conductance response and tracks physiological fear. In the reminded condition, fear didn't return during the follow-up test, while it did in the non-reminded condition. This suggests that reconsolidation-based extinction disrupts the fear memory trace itself, preventing the return of defensive reactions and potentially leading to more lasting changes in fear responses. Now for the changes in functional connectivity between the VMPFC and amygdala. Psychophysiological interaction, or PPI analysis, showed stronger coupling in the non-reminded condition compared to the reminded one. In standard extinction, this stronger connection helps create a new inhibitory memory, which is basically like a safety signal to suppress fear. In the reminded conditions, though, this weaker coupling suggests something different. Instead of building a new layer of safety, reconsolidation-based extinction seems to directly target and alter the original fear memory itself, potentially leading to longer-lasting effects.
Let's now discuss why these results are important in research on how memories of threats can be changed. These findings are groundbreaking because they address a major gap in our understanding, specifically how the prefrontal cortex is involved in weakening or erasing threat memories during the process of memory reconsolidation. By answering this question, researchers have advanced insights into how healthcare specialists can modify, weaken, or remove memories of fear, thus having direct implications for treating anxiety disorders like PTSD. For instance, since the results show that extinction during reconsolidation reduces the need for the ventromedial prefrontal cortex to manage fear responses, exposure therapy could be more effective if it coincides with the reconsolidation window of fear memories, and for people who struggle with anxiety, as the therapy would rely less on their ability to consciously regulate fear using the ventromedial prefrontal cortex. The results from this study fit within the larger literature because they advance the understanding of how fear-related memories can be changed and how different brain regions work together to help reduce fear. Results showing that the strong connection between the ventromedial prefrontal cortex and amygdala is important for getting rid of traumatic memories fits with previous research in anxiety disorders, showing that the ventromedial prefrontal cortex helps manage fear by regulating the amygdala. For instance, in the accelerator brake model of PTSD, subregions of the PFC, like the ventromedial prefrontal cortex, show impaired functioning, whereas the amygdala input is enhanced, resulting in exaggerated fear responses and difficulty in reducing fear over time. This suggests that the healthy coordination of these areas, specifically the ventromedial prefrontal cortex, enhances adaptive threat expression and extinguishes fear, as the current study shows. The results have also sparked some debate in the research community. Since these findings were made, many new studies have looked at using retrieval extinction techniques to change fear memories in both human and animals. Some studies have found a long-term reduction in fear responses, and some have not, both in humans and in rats. These mixed findings have led to questions about whether retrieval extinction actually changes the original fear memory or just makes it easier to suppress.
While the study offers valuable insights, there are a few important methodological limitations to consider. First, the sample size was far too small, with only 19 healthy participants. This limits generalizability, and especially to clinical populations like those with PTSD or anxiety, where prefrontal cortex and amygdala function may differ significantly. Second, the follow-up period was only 24 hours, and without longer-term testing, it's unclear if the effects of reconsolidation-based extinction are truly lasting. Third, the ecological validity is limited. Using mild electric shocks and visual cues works in a lab, but it doesn't fully replicate real-world fear or trauma, which may affect how the findings translate to everyday situations. And finally, there are some neuroimaging limitations. While fMRI provided valuable data on prefrontal and amygdala activity, it has limited temporal resolution, making it difficult to capture rapid neural processes like reconsolidation. Plus, fMRI measures blood flow and not the underlying neural mechanisms like neurotransmitter changes or synaptic activity, which might offer deeper insights. Addressing these limitations in future research could strengthen the findings and their clinical relevance. While this study reveals an exciting possibility, and thus that extinction during the reconsolidation phase may bypass the impaired ventromedial prefrontal cortex pathways seen in anxiety and PTSD. By directly modifying the threat memories instead of suppressing fear, this approach could offer more stable, lasting fear reduction and pave the way for more effective treatments.
you.